Okay. Hi, I think we can start the class. Hope my audio is working. I hope I'm audible and my screen is visible. If at any time I'm not audible or if the presentation is not visible, please let me know. So this is week 10 live session of NPTEL course, host pathogen interactions. And the course instructor is Professor Himanshu Kumar from ISER, and I'm Sumati Raviraj, TA from University of Hyderabad. Yeah. So we will be discussing this week's assignment. Few questions we will address directly. Few we can, just go through the topics so that it will be helpful for you to answer the questions because most of the questions are theoretical. So week 10, the syllabus that was covered was influenza virus and disease from part 1 to part 6, and then arbovirus and arboviral diseases, and arbovirus and dengue virus disease, infection, arbovirus and Zika virus infection. So hope you have watched the videos and understood the topics. So this session is to clear the doubts or to help you clear the assignment, and if you have any doubts regarding the assignment or regarding any of the topics, you can always mail me to this email ID. The doubts will be cleared via the mail or it can be discussed in the next session. So the first is overview of mosquito transmitted viruses. So we have Zika virus here, and then we have dengue virus and West Nile virus and then yellow fever. So Zika virus, it causes mild illness. Just a second. So it causes mild illness, but it is linked to congenital Zika syndrome and leading to birth defects and microcephaly, etc. And then dengue virus can cause severe hemorrhagic fever and shock syndrome, especially in reinfections. And West Nile virus is asymptomatic mostly, and it can lead to neuroinvasive diseases in some cases. And then yellow fever, it causes jaundice, hemorrhage, and organ failure in many cases. So these are mostly very uh, lethal diseases which affect people and have a lot of symptoms which are very which cause lot of health problems, complications, and there are also few which recur, and these kind of fevers are mostly transmitted via mosquito bites. So Zika virus, dengue virus, West Nile virus, yellow fever virus, all are transmitted via mosquitoes. That is, mosquito is the vector. So every virus has a specific vector species, even though all of them are transmitted via the mosquito. So the primary mosquito species for Zika virus, which hosts it, is Aedes aegypti and Aedes albopictus. And for dengue, it is Aedes aegypti and albopictus again. For West Nile virus, it is Culex species, and for yellow fever, it is again Aedes aegypti. So all these are vectors for all these kind of viral fevers that are transmitted and have even caused epidemics and pandemics in humans. Then, other than this, the other option in the question was Powassan virus. So, Powassan virus is not transmitted via the mosquito, it is transmitted via ticks. So, these are insects which are present on the skin or on the body. So, the vector, unlike others for Powassan virus, is tick borne, making it more similar to diseases like Lyme disease and anaplasmosis rather than mosquito borne infections. Then the geographical range or the area where it is more prevalent, the Powassan virus, is mainly in North America and especially in the US and Canada. Whereas mosquito-borne viruses like Zika, dengue are more common in tropical and subtropical regions. And the transmission speed of this is via the ticks that can transmit it in as little as within 15 minutes and after biting, and whereas Lyme disease transmission usually takes 24 hours. Then the severity, while it is a rare virus, but however, it is a very severe neurological complication and it can lead to encephalitis, meningitis, and it has a very high fatality rate than normal mosquito-borne viruses, which are common and prevalent in the tropical and subtropical regions. So the ticks, which is the vector for Powassan virus, the primary species are Exodus scapularis and Exodus cookie. These are the two species that are prevalent or are primary vectors for transmission of Powassan virus. The next is mechanism of severe disease in secondary dengue infections. So, secondary dengue infections can lead to more severe diseases such as DHF, DSS, and ADE. So, DHF is dengue hemorrhagic fever or dengue shock syndrome due to phenomenon called antibody dependent enhancement. So, both of it is because of the antibody dependent enhancement, and then also there is these are the secondary infections. So the primary infection is when a person is infected with one of the four dengue viruses, that is the serotypes 1, 2, 3, and 4, and the immune system generates neutralizing antibodies specific to the serotype that was infected. The secondary is that if the same person is later infected with a different serotype, the pre-existing antibodies may not fully neutralize the virus, leading to ADE, that is antibody dependent enhancement. So the primary is when obviously out of four, one of the serotypes will be infected. Then you have the antibody specific to only that serotypes. You won't, even though the antibodies could be similar to other serotypes, they are not completely complementary to them, so they are not very efficient during the secondary infection, which could be of a different serotype. So that is how you develop the secondary dengue infection complications. So if the same person is again infected with a different virus or a different serotype of dengue, then already the memory of the antibodies that was generated due to a similar kind of virus of a different serotype will again be produced, and it will help in neutralizing the virus, and this leads to ADE. Then antibody dependent AD, that is antibody dependent enhancement mechanism. How does this exactly happen is non-neutralizing antibodies facilitate viral entry. So even though these antibodies are produced, they try to neutralize it. The neutralization process is not effective because it's not the exact complementary serotype. So they will not completely neutralize. It will be inefficient, and this will lead to uh, this will help in facilitating the viral entry. So pre-existing antibody from the first infection bind to the new dengue virus, but however they fail to neutralize it because it's of different serotype and they're not entirely complementary, and they can, even though it is of similar type, they cannot completely neutralize it. So these antibody virus complexes, however, they interact, they bind to the virus, and these antibody virus complexes with the Fc receptors on monocytes and macrophages and dendrocytes. Then this promotes increased viral entry into the immune cell, leading to enhanced viral replication. So since they are able to now bind to the cells, especially the immune cells, but however they're not neutralized completely. So it gives them a free pass, or it gives them an entry channel into these immune cells, which leads to enhanced replication of these secondary infection viruses. Okay, so the increased viral load and immune hyperactivation occurs. So higher levels of viral replication leads to a cytokine storm, because when the concentration of the virus is too high, you need secretion of lot of uh, interleukins and pro-inflammatory cytokines. So it creates a cytokine storm with excessive release of pro-inflammatory cytokines like TNF alpha, IL-6, IL-10, and other interleukins. And this results in increased vascular permeability, because the cytokines then have to then attract the other immune cells to, you know, show their action at the site of infection. So when the immune cells have to travel, they increase the vascular permeability for their movement, and then plasma leakage occurs because this is excessive in this case, and then endothelial dysfunction occurs. So anything excess is obviously harmful. So due to the excess cytokine storm, uh, the vascular permeability is increased in a much higher rate, and due to this, your endothelial cells will be damaged in your capillaries and blood vessels. So this will eventually lead to plasma leakage and complete endothelial dysfunction. Next is complement activation and coagulation disruption. So the overactivation of the complement system. So we studied the complement system previously. We have the classical, we have the alternative, lectin activated. So all these are stimulated by the antigen antibody complexes, or by the and manus binding lectin, which binds to the carbohydrate. So all these, because there is now increased infection, amplified infection, enhanced entry, so this complement system also will be overactivated, and this will also contribute to vascular leakage and then hemorrhagic manifestations. Then along with this, platelet dysfunction and depletion also occurs. So this depletion and dysfunction of the platelets will increase the risk of bleeding and thrombocytopenia. So platelets are important for uh, preventing the bleeding and formation of clots, and stopping the bleeding at the site of injury. But due to these kind of overactivation of the complement system, the coagulation of the blood will be disrupted, because the platelets are now depleted or they are dysfunctional. So if there is no coagulation of blood, it will increase the bleeding, and then it will lead to thrombocytopenia. Then other than this, the severe outcomes are DHF and DSS. So the plasma leakage can then lead to hypovolemic shock, and increased vascular permeability causes organ damage and hemorrhagic manifestations. So this is why we have the severe complications, that is dengue hemorrhagic fever and dengue shock syndrome. So in the shock syndrome, it is hypervolemic shock due to the plasma leakage. Now your blood is void, or it has very less volume and very less plasma, and then you have hemorrhagic shocks to the organs, and eventually lead to organ damage. So to summarize this, risk factors of severe dengue. So the previous infection with a different dengue serotype, high viral load due to enhanced immune cell activation, strong pro-inflammatory response leading to vascular leakage, and then also along with this, the host genetic factors like the HLA variations, MHC classes, all these also will affect on how the immune system responds to this, and the age definitely, and also the immune status depending on the age, that is in children and in adults and in elderly, it is different. So mostly in young children and elderly people, the immune system is compromised, or it is still not yet completely functional in young children. So they are at higher risk of such kind of secondary infections and secondary infection complications. So this is about the diseases, severe diseases in case of secondary dengue infections. Just a second. Okay. Yeah. So the next question was regarding the influenza virus replication. So to prevent influenza virus replication, what are the mechanisms? So there are different mechanisms in the viral life cycle that can be targeted, and major steps that are involved in influenza replication cycle, and uh, once they are identified, the particular step or particular place can be easily targeted, and corresponding potential targets can be made, and antiviral interventions are then tested. So the major steps in influenza replication cycle and the corresponding potential targets for antiviral intervention are in the step of viral entry, that is inhibition of the viral entry. So at the entry itself, you can stop it. So the mechanism is influenza binds to sialic acid receptors on the host cells via hemagglutinin, that is HA. So this allows endocytosis, which will be a point of entry for your virus. So targeting this is the sialic acid analogs like your zenamivir, peramivir, etc. These are drugs that are used which will block the virus binding. Other than this, they can also use monoclonal antibodies against hemagglutinin, because hemagglutinin helps in the endocytosis process. So if you add the antibodies, monoclonal antibodies against the HA, so this will bind to the HA and it will prevent it from the function. So it will prevent it from attaching to the receptor. Then fusion inhibitors. So directly the fusion membrane will be targeted. So it will disrupt the membrane fusion in the endosomes, preventing the endocytosis, in turn preventing the virus entry. Then uncoating inhibition. So in this, after endocytosis, the virus will enter the endosome, that is the acidic endosome, and it activates M2 ion channel, which will allow proton flux and viral uncoating. So to target this, M2 ion channel blockers are used, which will prevent the flow of protons and which will eventually help in preventing viral uncoating. So the examples for this are amantadine and rimantadine. So these drugs help to prevent viral uncoating. Then these are, however, less effective against currently circulating influenza strains due to resistance. So as you know, virus always has genetic variations. It can, easily change its genetic information, and it can easily get resistance towards something, or it can change its method of uh, infection based on every infection or how it evolves. So currently, many influenza strains are resistant to this M2 ion channel blockers. The next step could be the viral RNA replication. So inhibiting this step. So the viral RNA dependent RNA polymerase. So the viral RNA needs the RNA polymerase for its replication. So this complex has PA, PB1, PB2. Uh, it transcribes and replicates the viral genome inside the nucleus. So if this is inhibited or it is targeted, then the replication, the RNA replication of the virus can be stopped. So to target this, polymerase inhibitors are used. Example, baloxavir, marboxil. So this inhibits the cap snatching by uh, targeting PB2, which is a part of the complex, and preventing viral mRNA synthesis. So here we discuss three steps which can be inhibited, that is the viral entry, uncoating, and viral RNA replication. So for viral entry inhibition, we have sialic acid analogs, that is the zanamivir and peramivir, and we also have monoclonal antibodies against HA, that is hemagglutinin. Then we have fusion inhibitors, which disrupts the membrane fusion of endosomes. We also have uncoating inhibitors, that is M2 ion channel blockers, amantadine and rimantadine. And then we also have viral RNA replication inhibitors, which is basically a polymerase inhibitors, which inhibits the RNA dependent RNA polymerase. So it targets the PB2 of the complex and prevents the viral mRNA synthesis. Next step inhibition is for viral protein synthesis. So the mechanism is the viral mRNAs hijack the host ribosomes to synthesize viral proteins. So the potential targets for this is host target inhibitors. So since now they are using the host machinery, what we have to target is the host machinery itself. So the ribosomes will be blocked, which will prevent the processing of viral mRNA. And then interferon stimulating agents are given. This enhances the host antiviral defenses. Then next step for inhibition is the viral assembly and packaging. So in this step, newly synthesized viral RNA, nucleoproteins, and polymerase components assemble into a progeny virions. So now everything has been synthesized. So now the new virions will be packaged. So this step can also be targeted at host chaperone level. So if the host chaperones are inhibited using the appropriate inhibitors, this will prevent the proper folding of the viral protein. So as it uses the host machinery now, from now on, that is from after translation, it uses the host ribosome. In the same way, it uses the host chaperones. So chaperones help in the proper folding of the proteins in the cells. So it also uses the chaperones for proper folding of its own proteins, that is the nucleoproteins, polymerase, and whatever proteins is required for it to be assembled into the virions. So if they are not properly folded, they will not be functional. So the chaperone inhibitors can be used to inhibit the function of the proteins. Then viral nuclear export inhibitors. So now this has to be exported. So they block the RNP transport, that is the RNA nucleoproteins and polymerase. Export means packaging into the uh, virus itself now, progeny virus. So this transport into the virus will be stopped. That is to block the transport out of the nucleus. So sorry, it will block the transport of it outside the nucleus. So it's nuclear export inhibitors. So this step can also be prevented using blockers and inhibitors to block the export uh, systems which it uses for cargo transport. Then viral release inhibition. So once it is now packaged and it is exported into the outside the nucleus, and now it has to come outside the cell. So that is the release of the newly formed virus particles. So now this should can also be prevented. The virus buds from the host membrane, uh, but remains attached via sialic acid until neuraminidase Na cleaves it, allowing release of the new virions. So once they are exported outside the nucleus, now these viruses will bud out of the cell. So it will bud out of the cell, but anyways it will stay attached to the sialic acid. So the sialic acid we saw before that it helps in attachment of the virus to the cell. So the same sialic acid will now also be helping in holding on the newly formed virus particles until neuraminidase enzyme comes and cleaves it. Now when the enzyme cleaves it, it will allow the release of the virions from the cell. So if you target neuraminidase, that is, if you use neuraminidase inhibitors like oseltamivir, that is Tamiflu, this is very commonly used, and zanamivir and peramivir, this will prevent the release of the virus from the cell and it will stop the infection cycle. So once if it's not released, it cannot now infect a new cell and it cannot continue the cycle. So it will stop the cycle basically. So the next three steps where we learn for the inhibition are the viral protein synthesis, which is inhibited using host target inhibitors for ribosomal blocking, and then increasing the production of interferon using interferon stimulating agents. Then in the next step, the inhibition was to prevent the viral assembly and packaging. So it will use the host chaperones to first probably fold the viral proteins before packaging. So that step itself can be prevented using chaperone inhibitors, and viral nuclear export inhibitors can be used so that the export of RNP out of the nucleus can be prevented. Then next step was the viral release inhibition. That is, once all the viruses are now formed, we can prevent it from releasing and infecting newer cells. So for that, the blockers used or the inhibitors used is against the neuraminidase enzyme, which helps cleaving the sialic acid bond between the virus and the cell. So this is how, and at every step there can be used, which can be used to, inhibit or stop the cycle of virus replication at any point, either from the point of entry to the point of exit, that is the release. So using different targets, different proteins, different enzymes as targets, using drugs against it, inhibitors against it, blockers against it, we can stop the virus replication. So influenza virus can be targeted at multiple stages, including the entry, uncoating, RNA replication, viral protein synthesis, protein assembly, and also release. So from the first step to the last step, every step can be inhibited using multiple targets, uh, multiple steps and proteins as targets. So the most effective antiviral drugs. However, among all these, the most effective antiviral drugs that are currently in use are NA inhibitors that target neuraminidase and RNA polymerase and M2 uh, ion channel inhibitors. So all these are inhibited or blocked so that they can stop the viral replication virus infection cycle from going ahead further and infecting new cells, and continuation of the infection will be stopped. So once the reproduction or the virus cycle is stopped, then there will be no more infection. So the next was Zika virus, and in which family it is classified. So the Zika virus is classified as a positive sense single-stranded RNA virus. So it has single-stranded RNA as its genetic material, and the RNA is a positive strength, sorry, positive sense single-strand RNA. So the positive sense a single-strand RNA virus belonging to the flavivirus genus within the Flaviviridae family. So the classification of the virus is as follows. The family is Flaviviridae, genus is Flavivirus, and species is Zika virus. That the genome type is, as we discussed, the positive sense single-stranded RNA. Wow, okay, this is like a tongue twister. So envelope, and it is enveloped lipid bilayer with viral glycoproteins And the morphology is icosahedral symmetry, like the virus capsid we know like a diamond shaped. Sorry, I did not provide with an image, but it is icosahedral symmetry and it is around 50 nanometers in diameter. And related viruses are dengue virus, West Nile virus, yellow fever virus, Japanese encephalitis virus, etc. The key characteristics, that is the transmission, is primarily via mosquitoes, because you can see the related viruses are also transferred via the mosquitoes. So that is the vector. So primarily via Aedes mosquito. So Aedes aegypti, Aedes albopictus, and it is also transmitted sexually, perinatally, and through blood transfusion. So there are multiple modes of transmission, but major mode is like majorly the more probability for you to get infected via this is by a mosquito, that is the Aedes mosquito. Other than this, it can be transmitted sexually, perinatally, and through blood transfusion. So tropism, that is, it infects the skin, fibroblasts, dendritic cells, endothelial cells, and also it has a strong affinity for neural progenitor cells. So that is why it causes microcephaly in fetus, that is, the size of the head will remain very small and then it will be non-functional and the babies usually don't survive. Then pathogenesis is, it leads to mild symptoms in most cases but can cause congenital Zika syndrome, that is microcephaly, and neurological complications, that is Guillain-Barré syndrome. So this is about the Zika virus. It belongs to the family Flaviviridae, genus Flavivirus, species Zika virus, and the genome type is positive sense single stranded RNA. Okay. And it is an enveloped virus. It is lipid bilayer and it has viral glycoproteins. It is icosahedral symmetry and it is 50 nanometers in diameter. Then it is similar to the dengue virus, West Nile virus, yellow fever virus, Japanese encephalitis virus, etc. And similarly to these virus transmissions, it also is transmitted via the Aedes mosquito, but it can also be transmitted sexually, perinatally, and through blood transfusion. It affects the skin fibroblasts, dendritic cells, endothelial cells, and it has a strong affinity for neural progenitor cells, which leads to congenital microcephaly, that is congenital Zika syndrome. So microcephaly leads to, having smaller head and smaller brain, and leads to neurological complications like Guillain-Barré syndrome. So this was about the Zika virus. Next is encephalitis. So which family predominantly include the arboviruses? So arboviruses are basically arthropod bone viruses. So they come under the families Flaviviridae and Togaviridae, and these are responsible for encephalitis. Then Flaviviridae family, just now we discussed, it has the genus of flavivirus, and many encephalitis causing arboviruses belong to flavivirus genus, and they are again transmitted by mosquitoes, but this is by the Culex species, and it can also be transmitted by ticks. The notable flavivirus, so this is, I'm talking about
Flavivirus.
notably does someone have any doubt? Did someone speak something over the mic? Uh, was there a question? Okay. So this here I'm talking about the Flaviviridae family, not particularly about encephalitis. Okay. So notable flaviviruses that cause encephalitis. Now we are coming to encephalitis. Our West Nile virus, which causes West Nile encephalitis primarily in areas like America, Europe, Africa, and also Asia. Then we have Japanese encephalitis virus. This is the leading cause of viral encephalitis in Asia. So you can see it's more prevalent in Japan. And then we have St. Louis encephalitis virus, which is an endemic to North America. So the other than this, we also have tick-borne encephalitis virus, which is transmitted by ticks. So this causes severe encephalitis in areas of Europe and Asia. Then we also have Murray Valley encephalitis virus, which is more prevalent in Australia and New Guinea. Then the other family is the Togaviridae family, which has the genus alpha virus. So some alpha viruses cause severe encephalitis mainly in the Americas, and notable alpha virus causing encephalitis are Easter equine, Western equine, and Venezuelan equine. So these are different types of viruses which cause encephalitis from the Togaviridae family. So among these, the Eastern equine encephalitis virus is highly virulent, and it has a fatality rate of more than 30%, and it can be as high as 70% in some areas. Then Western equine virus is less severe, but however, it causes neurological damage. Then Venezuelan equine encephalitis virus can cause epidemics in horses and in humans both. So this was about the Flaviviridae and Togaviridae families. The next is to determine which of the following is not a function of hemagglutinin protein of the influenza virus. So we discussed the hemagglutinin protein of the influenza virus, which helps in the endosome, the pH, right? 1 second. Yeah. So yeah, it helps in allowing endocytosis. So it binds to the sialic acid residues to allow endocytosis. So which among the following is not a function? So let's see what are the functions of hemagglutinin first. So first is the receptor binding, that is it binds to the sialic acid receptors on the host cells, initiating the virus attachment. Then next is it mediates endocytosis after binding. That is, the virus is internalized via receptor mediated endocytosis into endosomes, and then membrane fusion occurs. So yeah, at low pH inside the endosome, hemagglutinin undergoes a conformational change, exposing the fusion peptide. So now this will help facilitate the viral and the endosome membrane fusion. Then host tropism and immune evasion. So hemagglutinin determines host species specificity, that is human versus avian strains. It is the strains which infect the humans are different from those which infect avian, that is the birds, and is major target for immune responses undergoing antigenic drift and shift. So yeah, hemagglutinin is helpful in receptor binding, it is helpful in endocytosis, it is helpful in membrane fusion. So these are the major functions of hemagglutinin. What is not a function of hemagglutinin is during viral genome replication. So hemagglutinin does not play a role in replicating the viral RNA. And influenza replication, that is the RNA replication, is carried out by viral RNA dependent RNA polymerase. Like we discussed, the RNA dependent RNA polymerase, which has a complex with made of PB1, PB2, and PA proteins. So it is involved in the beginning steps till membrane fusion, and however, it is not involved in viral genome replication. Side note, it is also involved in determining the host species specificity. So this is about the hemagglutinin, that is the HA protein of influenza virus. Next, so why is low pH important for influenza virus fusion process? So we read that at low pH inside the endosome, HA undergoes conformational change. This exposes the fusion peptide, which facilitates viral endosomal membrane fusion. So low pH is crucial to trigger the conformational changes. So, protein conformations change depending on the environment, change depending on the interactions, change depending on the chaperones that are that sometimes fold them or reconfirm them. So if there is ion changes, pH changes, the conformation of your proteins will change so as to either activate or inactivate them. So in this case, the low pH helps the influenza virus in fusion. So how it helps is it triggers the conformational changes in the HA protein. So HA protein, when it is exposed to low pH, so that time it will undergo a conformational change, that is a change in the way it is packed or it is folded to expose the uh, fusion protein, a fusion part of it. So this allows the viral and the endosomal membranes to merge. So they fuse together. This will facilitate the genome release into the cytoplasm. So only when the membranes fuse, it can inject or it can release the its own GM into the host cytoplasm. So this is a very important step. So let's go step by step how this occurs. So the first is endocytosis and acidification. That is nothing but low pH. So after the influenza virus binds to the sialic acid receptors, it is internalized into the host cell via endocytosis, forming an endosome. So as the endosome matures, that is, as the endosome grows and becomes a mature endosome, the proton pumps, that is the VATPases, lower the pH to approximately 5 to 5.5. So the pH goes from around 7, 7.2, 7.4 to 5 to 5.5. So this happens when the endosome matures. So there is acidification or lowering of the pH that occurs. Then due to this, the HA changes its conformation. So the HA is initially in a metastable state. Then due to the acidic environment, uh, it protonates the key residues of HA. So since the proton pump is active, that means you have proton flux, which leads to lower pH. So this protonates the key residues in the HA protein. Then the HA1 to shift to it shifts and it exposes the HA2 part. So this HA2 part, it has the this HA2 portion of it, it has the residues or it has the peptide which helps in fusion. So usually this will not be exposed, but when this metastable structure undergoes conformational changes due to protonation, the HA1 shifts and exposes the HA2. So now this allows the fusion because it contains fusion peptide residues. So membrane fusion initiation then occurs. So the exposed fusion peptide of HA2 inserts into the endosomal membrane. So fusion is basically nothing but it will literally like fuse or join, or the uh, complexes will mix with each other, or whatever layer, membrane layer is there, it both will mix with each other. So the HA2 inserts into the endosomal membrane, anchoring the virus. So this holds the virus into the host. Now, this will help in insertion of the genetic material. So now the HA undergoes further structural rearrangement. So now after anchoring the virus into the endosomal membrane, further more structural rearrangements occur in the hemagglutinin protein. So this brings the viral and the endosomal membranes closer together. Then formation of a fusion pore occurs. So once the uh, virus and the endosome have been fused and they are attached to each other, they are close to each other, the virus is anchored on it. Now it creates a fusion pore. So the Ha2 trimer is a multimer, three monomers of Ha2 is a trimer. So the Ha2 trimer, so three Ha2s together fold into a stable 6 helix bundle. So this is a little detailed information. You may not need this, but it's just there. Okay, you can just remember it vaguely. You don't have to spend a lot of time on it. So the Ha2 trimer folds into a stable 6 helix bundle. So basically what happens is conformational changes. The trimer helps in changing the bundle shape or arrangement. So this forces the membrane into close proximity. So this will further pull the membrane and the virus together. So basically it is just changing its structure in a way so that it can pull the membrane closer, it can hold it, it can fuse together. So eventually it results in membrane fusion. So this now allows the viral nucleoproteins uh, to escape into the cytoplasm. So this is the export of virus from nucleus to the cytoplasm. Then viral genome release has to occur. So after fusion, the M2 ion channel helps acidify the viral interior. So now the viral genome has to be released. So the viral interior further dissociates into RNPs from the further dissociating RNPs from matrix, that is M1, allowing the transport of the nucleus for replication or allowing the transport of the genome for replication. So this is the step-by-step process of low pH fusion. So since pH plays a major role for the initial change of the HA conformation, which initiates the start of fusion, it is very important. Otherwise, the fusion will not occur, and then viral genome release will not occur, and the virus replication cycle cannot begin. So the low pH is essential for HA activation and membrane fusion. Without this acidic environment, the virus cannot escape the endosome, which will then prevent the infection. So that is why drugs like bafilomycin A1, which blocks endosome acidification, can inhibit influenza virus entry. So in the fusion step itself, if you can inhibit it, then there will be no virus entry, there will be no viral genome entry further, and you can prevent the infection. So drugs like bafilomycin can inhibit virus entry. Then next is drug targets that uncoat drugs that target the uncoating and replication of influenza virus. So there are several drugs which target the uncoating and replication steps. With, so these include, like few of them we have already discussed before. So these include the M2 ion channel inhibitors, which prevents the uncoating, and then the RNA polymerase inhibitors, which block the replication. Then amantadine is the most relevant antiviral drug that prevents influenza virus uncoating, while chloroquine has potential antiviral properties that interfere with the endosomal acidification. Then, particularly amantadine, it prevents uncoating. How it does is we already discussed. Amantadine blocks the M2 ion channel. in the influenza A virus, preventing the acidification of the viral interior. Then this stops the viral uncoating and it prevents the release of viral RNA into the host cell. It is effective against influenza A, however, but not influenza B. So there are a few limitations of it. It's high resistance in circulating strains because it undergoes change. Influenza virus is very dynamic. It keeps undergoing change. That's why we keep, getting flu every now and then. So it's highly resistant in the circulating strains. So currently it's having very limited use for treatment. Then chloroquine, potential endosomal acidification inhibitor. So chloroquine raises the pH of the endosome. So it prevents the acidification. So the hemagglutinin does not become active and it does not start the fusion process. So this may disrupt the fusion of some viruses that rely on low pH for membrane fusion. However, its effectiveness against influenza is not well established when compared to other antivirals. So, amantadine is a known influenza A antiviral that prevents uncoating. Chloroquine is some potential antiviral effects, but it is not well established, so it is not primary treatment for influenza. And better alternatives for influenza treatments are baloxavir marboxil, which blocks the viral RNA replication, key step, and oseltamivir, that is Tamiflu. This is also very widely used and it prevents the viral release. So other drug pairs that were given in the options were penicillin and amoxicillin. So penicillin, amoxicillin, they are antibiotics, so they will not be affecting the virus. So these are effective against only bacteria. Then also we have acyclovir and ganciclovir. So these are anti-herpes virus drugs targeting the DNA replication in case of herpes virus and not influenza. Other than this, there was aspirin and ibuprofen. So these are NSAIDs. So they are used for relief purposes and they do not affect the viral replication. So they are basically given for pain management. So they are to just reduce the symptoms but not to prevent, or it does not anyway stop the viral infections. So they are non-steroidal anti-inflammatory drugs. So basically it reduces the inflammation, it relieves the pain and lowers the fever, so headaches. These are common painkiller drugs that are used. They are only for symptoms. Then ritonavir and saquinavir. These are for HIV, that is HIV protease inhibitors. They are not related to influenza replication. So, in all the options given, it was amantadine, if I'm not wrong, is the answer for it. Then next is the influenza virus genome has the following key characteristics. So the genome type of it is segmented. It is 8 separate RNA segments, we know. So it is a segmented genome and it is a negative sense single-stranded RNA. It belongs to the family Orthomyxoviridae, and this is regarding the genome type, that it is 8 separate RNA segments. It is a negative single-stranded RNA. It belongs to Orthomyxoviridae. And other than this, the segments and their function. So each segment, why 8 segments? So each of the 8 segments of the RNA encodes for the essential viral proteins. So first is the PB2, second is for PB1, third is for PA, and fourth is for HA, fifth is for NP, sixth is for NA, seventh is for M1, M2, and eighth is for NS1, NS2. So the first segment that codes for PB2 is the function of it is As you know, it's part of the complex of RNA polymerase. So it is involved in cap snatching. This is one of the steps in that. Then PB1 is also a subunit or a part of the complex of the polymerase, and it is essential for viral RNA replication. PA is also part of the subunit. So these three are subunits of the polymerase. So PA cleaves the host mRNA for transcription. Then HA hemagglutinin we just studied. It binds to the sialic acid receptors, mediates entry and fusion, and then NP, that is the nucleoprotein. It encapsulates the viral RNA. It aids in nuclear import. So basically packs it. Then NA, that is the neuraminidase, which is important for release. That is, it cleaves the sialic acid residue, which uh, anchors the viral newly formed virus that is now exposed as virus bud. So it allows viral release from the cells. The seventh segment encodes for M1, M2. These are the matrix proteins. So M1 matrix protein. It stabilizes the virion. M2 is an ion channel, so it helps in uncoating. And then NS1 and NS2, they suppress immune responses, and NS2 is a NEP, that is for export. So it exports the viral RNPs from the nucleus. So they are nuclear export proteins. So these are the different proteins that are encoded by different segments of the 8 segmented genome of influenza virus. Then the site of replication, influenza virus replicates in the host cell nucleus, unlike most RNA viruses that replicate in the cytoplasm. So influenza virus enters the host cell nucleus and then replicates there. Most of the RNA viruses replicate in the host cytoplasm. So this uses a cap snatching mechanism to steal the host mRNA caps for viral transcription. So this is how it steals the uh, transcription mechanism, or it activates its transcription mechanism, and that is the reason it has to enter the nucleus. Then genetic variability. Segmented nature allows a reassortment. This is very important. So this leads to antigenic shift. So this is why it can cause pandemic. So because it's 8 segmented, these 8 segments are separate, so they can rearrange in any order during the course of time. So first time, if your influenza virus, that is the reason we don't have effective drugs. So if the first time the influenza virus has infected, the order is like 1 2 3 4 5 6 7 8. So next time it can be the same virus, and you had taken the drug for the first virus, which was effective, but the second time when the same virus has infected, it is not effective. That is because there is no change in the genetic material, but the order of the segments have now changed. So there is an antigenic shift. So now the order could be anything. So there are different combinations that can occur in the 8 segments. So it can be 1, 2, 4, 5, 6, 8, 7. It can be 1, 3, 4, 5, 6, 8, 7, 2. So like that, there are so many different combinations that can be possibly done. So that is the reason there are no effective drugs, and once you're infected, you have to wait through the time that your immune system and whatever drugs will take its time. For different infection, it is always a different time. So it undergoes antigenic drift also. So other than antigenic shift, it also undergoes antigenic drift, that is point mutations. So even a single codon change, uh, a nucleotide change, sorry, even a single nucleotide change can lead to change in the complete immune response against the virus. So it undergoes several point mutations, which leads to antigenic shift, leading to seasonal flu variations. So seasonal flus is because of antigenic shift, because sometimes uh, due to the that is why we get seasonal flu, that is from one season to another season, because of just a change in a single nucleotide. Now it can become more effective in particular season. Then virus types: influenza A, B, C, and D exist. A and B are most clinically relevant. Influenza A causes pandemics due to antigenic shift. So, influenza A is highly uh, uh, efficient because can it has it has more probability for antigenic shift. Then next was to find what all the statements about dengue are correct. So we'll go one by one for this. The first was dengue fever is caused by a virus transmitted exclusively by mosquitoes. So this is true. It is by the Aedes mosquitoes. So the dengue fever is caused by dengue virus, which belongs to the flavivirus genus, and it is transmitted exclusively by Aedes aegypti and Aedes albopictus mosquitoes. So this statement is correct. Next is infection with one dengue serotype guarantees immunity with other dengue serotype. This we discussed that it is not possible. So this is why the secondary infections occur. So this is false. So there are four distinct serotypes of dengue: one, two, three, four. Infection with one serotype provides lifelong immunity to that specific serotype only. That is, if you're infected with dengue 1, dengue virus type 1, so the next time dengue virus type 1 infects, you will be immune. You won't get the infection. But the next time dengue virus type 2 infects, you will be infected because it's a different serotype and the antibodies are not efficient enough or not complementary enough to neutralize it. But only temporary cross protection against others. Then antibody dependent enhancement, that is ADE, can lead to severe manifestations during secondary infections. That is true. We read about it. The DSS and the DHF are caused because of ADE. So ADE occurs when non-neutralizing antibodies from prior dengue infection enhances viral entry into host cells. This leads to severe dengue, that is dengue hemorrhagic fever, dengue shock syndrome, etc. Then they have their own symptoms and complications. Then next statement was, so this statement is true, and the next statement was early detection methods for dengue include RT-PCR and serological antibody tests. This is correct. So RT-PCR helps detecting the viral RNA levels, and it is effective during the first 5 days, 5 to 7 days of infection. Serological tests are for the antibodies. So IgM and IgG ELISA is done. So this will help confirm past or recent infection. So if the antibodies are already present, it's a past infection, and if it is in low number or if they are not present but RT-PCR is on, it's a recent infection. And also it helps know which serotype is infected. Then there are currently no effective vaccines available for dengue. So this is false. So dengue vaccines are given. Dengvaxia, it is approved dengue vaccine, but it is only recommended for individuals with prior dengue exposure due to the risk of ADE. So this is very interesting because, like how after the first infection, in the secondary infection you have the risk of non-neutralizing antibodies, in the same way, if you are vaccinated without previous exposure to dengue at all, then it will also lead to risk of ADE because they can also become non-neutralizing sometimes. Then again, it will lead to hemorrhagic fever and shock syndrome. Then there are also newer vaccines like TAC not not 3, Qdenga. So these are being developed and introduced in some regions. So they are coming up. So this is a false statement. Then supportive care is mainstay for treatment of dengue fever. So this is true. No specific antiviral treatment is available. So because of um the serotypes and its virus. So supportive care includes hydration, pain relief, medications, and monitoring the severe syndromes. Then, so that is why we also sometimes hear that there are also several deaths due to dengue because there is no specific effective treatment. If the symptoms are controlled, then it has to be cleared by our immune system itself. So then rising global temperatures have no effect or impact on dengue transmission. So this is also false because climate change and rising temperatures increase mosquito breeding. So this is now more favorable for the mosquitoes. Then if there are more mosquitoes, there is more virus replication or there is more uh, carriers for the virus, and then obviously the rate of infection will be more. So the climate change and rising temperatures increase mosquito breeding. That is why they are also very prevalent in the tropical and subtropical countries. And the virus replication, expanding dengue transmission area. So even in the areas which were earlier not affected with dengue because of the rising temperatures, even they will now start getting affected by dengue because of the shift or the breeding of mosquitoes in that particular areas. Usually in cold countries, there won't be mosquito population. But if the temperature increases over there due to global warming, then obviously the mosquitoes can start breeding there also. Then community awareness plays a vital role in dengue prevention and control. Of course, this is true. Then public health efforts, mosquito control, eliminating breeding sites, and educating people of how dengue is transmitted, how to safeguard yourself from mosquito bites, and not create an environment where the mosquitoes can breed, like proper sanitation, hygiene, uh, uh, and avoiding stagnant water near the houses or in the houses. So all this help eliminating the mosquito breeding or reduce the mosquito population. So these are crucial for reducing the dengue cases. So dengue fever is caused by a virus transmitted exclusively by mosquitoes. Yes. Antibody dependent enhancement can lead to severe manifestations during secondary infections. Yes, and early detection methods include RT-PCR and serological antibody tests. Yes, and supportive care is mainstay for treatment of dengue fever. True. Community awareness plays a vital role in dengue prevention and control. Also true. So all these statements are the correct statements regarding dengue virus transmission and diagnosis and treatment and care. So this was about the dengue. So that is it. I hope I have covered all the questions. So if you have any doubt in any question or any other topic, you can mail me on this email ID. So do you guys have any question? Okay. So we can end the session here. Thank you for attending.